Brought to you by Curious Moranland. Science literacy will make us great if we increase it. So what I want to do is talk about my genetics introduction. So for my students, at various points, pause the video. I'm going to be working through some key terms. Some of these are key terms that you're going to use in a variety of different ways. Like these terms. I can tell from the way the video is going, where it says test cross here, underneath is the word Punnett square, so I'm going to need to modify that a little. These are a list of questions, so pause the video for a second and see if you can answer these questions. By the time we take our test, you should be able to answer these questions. For example, when can you look at a dominant and tell its genotype? We're going to say no. But you can predict how by knowing about its parents or looking at its children. Like this question here, if a dominant and a recessive produce all dominants, that tells you something about it. If a dominant and recessive produces dominants and recessive, that tells you something else about it. We're going to start with classic Mendelian genetics, so we're going to deal with complete dominants, not incomplete, but we'll get to it. So by the time my students take this test, these setup questions should elicit a whole bunch of discussion and explanation, not just regurgitate an answer, but say why something is correct. So Mendel is where this is based on. So these are his three principles: segregation, independent assortment, and dominance from recessive. So we're going to start first with dominance from recessive. Segregation is actually easy because before doing this unit, my students learned about meiosis. We learned about homologous chromosomes separating. And that's really all we need to know. He didn't even know about that. So I'm not going to spend a lot of time going through the history of Mendel. I'm just going to go right into explaining his concepts. Mainly dominance from recessive. So let's first make sure we understand what a trait is. So I'm going to put these videos up soon. You should pause at various points to get the copy down some pointers that I'm going to underline and highlight. So this is the hardest thing to understand. The students really got to understand this. They really need to understand what exactly does it mean to say a trait versus a variation of a trait. So if we look here, basic definition. It's an alternate version of a gene. So what's a gene? If you look over here, here's two homologous chromosomes. That means they're from two different parents, but they're for the same size, same shape. They are for the same trait. So here's the deal. The trait is going to be the color. Okay? The variation would be purple or white. So a person can have two purple genes. They can have, and we'll just use a capital P. All right? A person can have, when I say person, I mean a flower. They can have these. The letter designations we use to help us better understand and build a whole construct. Now, this organism, can have a purple and a white, but I'm not going to write white as a W. I'm going to write it as a lowercase because I'm going to presume that it's recessive. So I'm going to go back here, and instead of writing white, I'm going to write little t's. Now, here's the deal. You can have two of the purples. You can have two of the whites, or you can have one white and one pink. I should have said purple. But we don't write 1, 1. We would just write it like this. And so ignore the numbers. I'm not doing a Punnett square yet. We'll do that later. So to elaborate, I've used this a couple of times. So let's say this is your paternal and this is your maternal. On here are genes. At the same line, which I'm trying to do, let's say I'll call that big A, that means this would be a little a. Those are variations. Again, a allele is a variation. It's a possibility. Back to the last slide here. I want to get rid of the ink and start kind of clear here. Okay. I said that the trait was color, but purple is an allele and white is an allele. All right? They are variations. So let's say we have a trait here, and this chromosome could have maybe 2 genes on it. My letter designation is just showing the possibilities, the alleles. Here's another picture, and it's trying to show, look at all these different loci. That means location. Homologous chromosome means, homo means same, logos means location. Same gene location. So if you look through here, you'll see it can be double big letters, double little letters, or one of each. Later on in this video, in the PowerPoint, you'll see the term homozygous, homozygous, heterozygous. So pause the video for a second, and when you're looking at the other terms, come back to this one. So we're going to skip through this, and all right, this is pretty wordy. Let's see. We're now going to introduce where Mendel's idea of dominant and recessive comes in. See, if you have 2 alleles for one trait, 2 possibilities for one trait, what that means is you can have 2 traits that are the same. So that's one combination. Another combination would be the other 2 traits, or one of each. When you have one of each, that's where this concept of dominance comes in. The one that's fully expressed is dominant. The other has no noticeable effect. It's there. And here, let's put a little star by this. At this point, we're going to presume complete dominance, and if you can see the recessive, you can know its genotype. You can't look at the dominant and know its genotype, but you can know it's dominant by the fact that you can see it. All right, pause the video and come back to this. So for my students, I normally am going to have them copy down genotype and phenotype. So I'm just going to throw those up there really quick. Here's a little bunny. All right, what is phenotype? It's the physical expression of genes. Start associating genes that are expressed as something tangible, as the trait that you're actually going to be measuring, the outward appearance. You'll notice BB is a genotype, but we need to always identify what the alleles are. So when we do Punnett squares, we're going to talk about identifying the alleles and designating a letter and then doing crosses. So genotype, oops, let's go back to that. That's genotype. Let's pause the video for a second. So after copying this down, if you're my students, go back and compare the written text that you see over here and here. It's pretty much the same information, but I believe that you should be able to look at something in a chart form or a paragraph form. So I'm going to go ahead and pause the video, or I would pause the video, write this down, then read through this and compare the genotype and phenotype description and the homologous, I should have said homozygous or heterozygous descriptions. So I'm actually going to move on back to our list of words. Let's talk about generations. So here's what we're looking at. The P represents the very first time you're measuring these genes. Their children would be called the F1. And Filial is the abbreviation, is abbreviated as F. And then, so they're the F1 generation. The F2 would be the second generation of children. Now, what's a simpler way of writing this? So P, hopefully this will show up on the screen. So this is the parents. The first Filial would be the first offspring. meaning the children. That makes F2 the second offspring. Now, not the second offspring of the P, but the second generation of offspring. Or, in parentheses, parents, children, in relation to the P, the grandchildren. And I'm going to run out of space, so I'm writing the word grandchildren here. So to close this video, let's go ahead and look at this. All right, so I'm going to label this the P. That means the offspring here are the F1, F1, F1. This is one parent, parent 1. This is another parent. And actually, I shouldn't write parent 1 and 2. I don't want you to confuse the numbers. We don't use the numbers for parent. We just say P parents, all right? And I'm going to just get rid of that. All right, so let's go ahead and further close this up by just advancing the slide a little. So what is it? It's mating. The parents and their children are the first filial generation, the children. Next, let's take the children and keep crossing them. Pause the video and copy down the definition of Punnett square, which is right here. We're not going to do it in this video. I'll elaborate later on Punnett squares. So let's look at it like this. This is the P generation. That means these are the F1s, 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 F1s. Now watch. The F1s become parents now. The F1s become parents. That makes inside this box, that is your F2. Okay, we're not going to do dihybrid yet, so I'm going to go ahead and close the video. And just remind you, this is brought to you by Curious Moranland. And thanks for watching. And this is my classroom because I didn't shut it off in time. So.